Thank you for showing up here right before lunch. Did tweak the title slightly just because didn't want anyone to have the mistaken idea that we're finished with this particular data set, but hopefully after 15 minutes I can convince you that it's a study system worth pursuing. So we've known about epizoic diatoms for a while. Decades and decades ago, we've known about ceticulous diatoms growing on cetaceans, whales, and dolphins. Some of the more recently described taxa we've also found on animals a little closer to shore with sea turtles and manatees. So in particular, two taxa that are going to show up a lot during this talk are Tertiocula and Trypterion. They tend to be pretty species depauperate. Most of these are monotypic, but in recent years we've found a lot more diversity, particularly in these last two taxa there. So Tertiocula in particular, Tom Frankovich spearheaded some great work on Tertiocula diversity on manatees. These are characterized by these very distinct pseudosepti at the center of the valve. And another interesting aspect about the manatee Tertiocula is that many of the ones we found are apochloritic. They're non-photosynthetic. Trypterion, a lot of work here, not just on the genus itself, but also some recent expansion of the Trypterion complex, new genera around this genus, Pulinia and Delnicola, that Roksana Majewska and her group worked with. Tom Frankovich again with Medlinella, and Catherine Rio-Gobon has described a couple of more species within these genera as well. Yeah, great. So what? Diatoms grow everywhere. Of course, that grow on animals. Why am I keeping you from lunch to talk about this further? We've heard about it a little bit already at this conference. We've seen some, or we'll see some great posters about this tonight and going on. What's the big deal? And what's interesting to me is the potential for these systems to look at dispersal and diversification of diatoms, particularly marine benthic diatoms, across the globe. There's this prevailing wisdom that anything smaller than a millimeter has unlimited dispersal, right? Everything is everywhere and environment selects. And okay, maybe we buy this for planktonic species. They get picked up by wind, they get picked up by currents. They have effectively unlimited dispersal. But what if you're a benthic diatom, like this beautiful little weed, Pseudictia dubium? You're 15, 20 meters down, you secrete a lot of mucilage specifically to attach to structures. How are you getting around? Okay, we've got models of storm surge and maybe just further dispersal by animals. I was going to discount storm surge as a stochastic event, which would be difficult to model, but listening to the plenary this morning, I guess we've got stronger waves and stronger storms ahead in our future, so maybe that's a less stochastic model than I thought. And if everything is everywhere, where are new species coming from, right? If there's no restriction to reproduction, then how do we get new species of these diatoms? And if we have epizoic diatoms that are restricted to just living on these animals, that gives us a great little system to study how these things, how we get population structure, how we get genomic differentiation from systems where it's not as stochastic, where we only get, where we know we get reintroduction and potential reinforcement of these populations every time the animals come together to reproduce themselves. And then, of course, the people who study these animals are interested in the diatom assemblage that we find on them, particularly because they think they might be useful for helping to identify where these animals have been. If the assemblage is tied to various local environments, then maybe we can tell a little bit more about where these animals are going based on the diatoms we find there. And as part of that, they've been very excited about the possibility of metabarcoding. We've heard a lot about that today. I guess if the animal people are really interested in metabarcoding, so they don't have to talk to diatomists anymore. And there have been a couple of metabarcoding studies on these epizoic diatoms already. The Rivera 2018, and then of course we heard Sunčica yesterday talking about some of the preliminary work in the Mediterranean. And that's great, that's wonderful, but there is certainly a limit to this barcoding study. For example, in both the Rivera and in Sunčica's work, we didn't see any of these putative obligate epizoic taxa, Tertiocola, Melanella, Trypterion. We didn't see any of these in the data sets. Why? Because data doesn't exist in the databases that we have already for these diatom metagenome, the diatom barcoding surveys. And in fact, we don't have a lot of marine benthic diatom data at all. So that limits what we can say about the metabarcoding results. And I thought it was particularly interesting that Rivera chose to focus on small nitzias, cryptic diversification in small nitzias on these turtles, when I would wager a kidney, you could go to pretty much any marine benthic environment, sample it, and find an undescribed small nitzia. So I don't think there's anything particularly exciting about that. Now, even if we don't have specific information for specific diatoms, maybe we can use the molecular phylogeny to sort of narrow down what these undescribed, unsequenced diatoms, where they might be in the unidentified barcodes, right? The problem there is, well, what kind of diatom is Trypterion? What kind of diatom is Tertiocola? They sort of fall into this broad category of morphology as gomphonemoid, heteropolar, septate. But the problem is that occurs in a lot of different places across the diatom phylogeny. We know that already. But a couple of authors have at least given us some places to look. Holmes suggested that Trypterion and Tertiocola are probably part of the Reucospheniaceae. We do have some Reucospheniaceae sequences in the database, so maybe any undescribed diversity around Reucosphenia is our putative epizoics. Now, if we're going to develop this system, there's a couple basic questions we need to answer. One, is the epizoic habit are even real, right? Diatoms are opportunistic. We know that there are some opportunistic diatoms that just happen to be growing on the turtles because or the manatees or the whales because it's a solid surface within the photic zone for large periods of time. So how many of these taxa are specific to these environments and how many of these are opportunistic? Can we even create a DNA library of these taxa? If they only grow on turtles and whales and manatees, do we have any hope of getting these things in culture at all? Or are we stuck with single cell work, which is possible? That's what we did with the Frankovich Tertiocola study, and it was not cost effective. It was a pain in the butt, and I don't have time to complain about it here, but you can find me afterwards with a beer and I'll happily complain about it. And then the third aspect is there even any genetic variation within these epizoic taxa at all? Can we detect any population structure, or is everything really everywhere? The Pseudictia dubium I showed earlier, where we have cultures from across the globe, they're genetically identical. We found no population structure to those yet. And so do we have a similar system here? So part of that is attempting to get these diatoms into culture. We've already seen Sunčica talk a little bit about sampling these guys, the brushes. Tom's been doing yeoman's work going across Florida, getting us plenty of samples from manatees and turtles. A portion of those samples get sent to me in Austin, and I spend hours and hours in front of a microscope picking them. And then, of course, recently we've had more efforts with culturing with Sunčica in the Mediterranean and Roksana in South Africa as well. And they've been largely successful. We've been able to get lots and lots of strains from these environments into culture already. Some of them are pretty clearly not specific to the animals, Neodelphinese, Starophora, but some of them are putative epizoic taxa, like these Pushkinia and our Trypterion complex, or this Agnanthes elongata. So, where do we find these in the molecular phylogeny? So we thought we might find them in Reucosphenia, down here, all on its own. They're not particularly close to that. So another marine gomphonemoid up here, Gomphonemopsis cystrodophaeodactylum. The pseudogompphonema is in the Naviculaceae, way off the chart there. But then our putative epizoics, Tertiopula and the Tritarion complex, are down here in this other clade with the Staranice and Praticula and some monoraphids. And I understand, yeah, there's a lot of poorly supported nodes between these, but if we do constrain all of our marine gomphonemoids into the same clade with Rheucosphenia, we result with a significantly worse tree. So these are not monophyletic, these are not part of the same clade. And really, there's no reason we would be looking for these taxa in the molecular clade where we're finding them. Staranice, Prashtinia, Fistulifera, Astardiella, and Schizostauron. There's no morphological feature that we would look at those taxa and say, oh yeah, we completely expect Tertiopula and Tripterion to be there. So that sort of underscores the importance of having these reference sequences in our database for metabarcoding. So yeah, in terms of the variation within these groups, some does exist. So again, this is not barcoding data. This is complete sequence data for SSU, RBCL, and PSPC. And in this phylogram, we do see some substitutions between the various groups. So with our Tripterion complex, with Fulinia from South Africa, from the Mediterranean here, from Florida, there's some distance between there. Only a few Medullinella, really only one Medullinella at this point. And then our Pelnicola caribbeana complex, again, we have from South Africa, and we've got quite a bit of variation between the Caribbean and Florida. So there is some genetic variation there that we could potentially mine for some fine-tuned population genetic studies. And it gets even better with Echinanthes elongata, where we even have potential host specificity here in a population of manatees, and then the various turtle samples for Echinanthes. And actually, one thing that I forgot to put in here goes back to Tertiopula, where we see the non-photosynthetic Tertiopula so far seems to be constricted to the manatees, and then Tertiopulas growing on the turtles do seem to have functional chloroplasts. So there's some sort of differentiation going on there. Are these epizoic habitats real? Diatoms are great to work with because they're microbes with morphology. The microscope is still a powerful tool. So there are quite a few people in our group that are still looking at all these samples with the light microscope. And what we see is, yeah, I mean, there's a lot of opportunistic taxa there, but there are some of these genera that we consistently see regardless of the location, the geographical location of these animals. And the nice thing about working with Florida is that we have an actually pretty decent, well-characterized flora of seagrasses in Florida Bay, where a lot of these manatees and turtles occur. So to wrap this up as quickly as I can, are epizoic taxa even a real thing? Yeah, it sort of looks like they very well might be. Can we create a DNA database to study these things at a much finer scale, both from a metabarcoding approach and from a population genetics approach? Yeah, they're going into culture pretty well, which invites a whole different series of questions, because if we can get these things, grow them away from the animals, why do we only find them on the animals? What's the source of differentiation there? What's the source of diversification? And then it does look like there is some genetic variation between various populations of our putative epizoic taxa. So that's something we hope to mine going forward. And thank you all for putting off your lunch to listen to this. Thanks to all our collaborators. And I just want to do my quick little advertisement that one giant you will notice is missing in all of this are the whales. We don't have whale diatoms yet, and there's a good reason for that. We don't have access to a lot of whales. If you do, you know anyone who does, please give us shoot us an email. We're very collaborative. We're very friendly people, and yeah, just you all know how to find me.
Thanks very much. I think we'll skip the questions now. Max explained that a beer is all that is